Hello, everyone. In today's lecture, we will cover another aspect of real-time PCR analysis, and this is the normalization of the obtained data. So to briefly summarize again, the qPCR workflow looks as follows. First, you will perform an RNA extraction, then you will remove any residual genomic DNA. You will then generate cDNA from the messenger RNA through a process called reverse transcription. And subsequently, you will perform the actual qPCR reaction to compare the expression of a certain gene of interest between different samples. And in the consequent data analysis, it is very important that you normalize the data that you obtained. And in today's lecture, I will explain to you why this is important and also how this works. So why is normalization of qPCR data very important? Well, because there is a lot of variability in qPCR experiments. First of all, you have the specific or biological variation. And this means that there are differences in transcriptional activity between different conditions or different samples. So these are actual differences in gene expression. And this is what we're interested in. So we want to know whether the expression of a certain gene that we're interested in, we call this our gene of interest, whether the expression of this gene differs between different samples. However, unfortunately, there is also a lot of non-specific variation, and this is what we call technical variation. And these could be differences in the amount of starting material. So let's say that you would work with plant samples, then it's possible that one sample is much larger than the other sample. You could also have large differences in RNA concentration, in RNA purity, and in RNA integrity. There could be different efficiencies of the reverse transcription reaction. So the cDNA synthesis could not be, could have different efficiencies between different samples. There could also be PCR inhibitors present in the samples, and the amount of these PCR inhibitors could also differ between different samples. And also the qPCR amplification might be different for different samples, for example, because inhibitors are present. So as you can see, the part that we're actually interested in is here, this biological variation. But unfortunately, there is also a lot of technical differences between samples that can influence the results that you obtain. And therefore, data normalization is very important to make sure that the results you obtain are accurate. So indeed, as is mentioned in the conclusion here, qPCR experiments are highly sensitive to technical variation, and therefore normalization is very important. So how can we minimize technical variation in qPCR? We can do this at different levels throughout the qPCR workflow. So as I mentioned, first of all, when you start from a sample, you will isolate RNA from that sample. And to minimize variation in that step, we always try to make sure that sample sizes are similar. So that, for example, when you work with plant samples, that all plants that you harvest are more or less the same size or weight. Then you obtain RNA samples. Next, we will generate cDNA from this RNA. And to do so, we will use the same amount of RNA input for each sample. So we will make sure that all our RNA samples are diluted to a similar concentration prior to generating cDNA from them. And then with the obtained cDNA samples, we will then perform the actual qPCR reaction. And when we obtain the data, we will normalize them, as I mentioned. Now, the next question is, how can we normalize qPCR data? And as you can see here, there is a large list of possible options. So you can normalize against sample size, against the total amount of RNA. You could spike samples with external RNA. You could look at the expressed allele repeats. You could use housekeeping genes for normalization, or alternatively, you could use validated reference genes. And we will now briefly discuss each of these possibilities and also their advantages or disadvantages. Starting off with sample size. So this is one of the potential normalization methods. So normalizing against sample size means that you will take into account the weight or the volume of the tissue that you used, the number of cells that are inside the sample, or something else. An advantage of this is that this is quite reproducible. So you can weigh, for example, each of your samples and then normalize against that sample weight. However, this is not always possible. Let's say you're not in the ability to weigh the samples or to count the number of cells, and then this is, of course, a problem. Because indeed, as I mentioned here, determination of dry weight or counting of cells is sometimes difficult. When using this normalization method, you do take into account differences in transcriptional activity between samples, which is what you were actually interested in, because this means that you will look at differences in gene expression levels. However, this method does not take into account potential differences between RNA concentrations, RNA purity, and RNA integrity between samples. It also does not take into account that the efficiency of cDNA synthesis might differ between samples, and also the presence of PCR inhibitors is not taken into account. So in conclusion, this is an unreliable method for data normalization. Another possible normalization method is to normalize against the total amount of RNA. So when you determine the RNA concentration using the nanotop, you could use this obtained value to normalize the qPCR data. However, this is also not always possible. Sometimes you might not have a nanotop available, you're not able to determine a concentration of RNA, then this is, of course, an issue. And again, even though you could normalize against the total amount of RNA, this does not tell you anything about the RNA purity and integrity, about the efficiency of cDNA synthesis, about the presence of PCR inhibitors between different samples. And also, what's important to keep in mind is that the total amount of RNA, which is what you determine using, for example, the nanotop, is not always representative for the messenger RNA fraction. And in qPCR, we are, of course, only analyzing gene expression, which means mRNA levels. So again, this method is unreliable for data normalization. Another possibility that is sometimes used is spiking the obtained RNA samples with an external control. So this means that a known amount of artificial RNA is added to your RNA samples. The advantage of this is that in this case, this will also correct for reverse transcription and differences in enzymatic efficiency. So this means that when cDNA synthesis is more efficient in one sample than in the other, this will be the case for the external RNA, the artificial RNA that you added to the sample, as well as for the RNA coming from your original sample. However, in this case, an equal amount of sample input is still assumed. And again, this does not tell you anything about the RNA purity and integrity of your samples. So also not the most ideal method. Another option is to use so-called allele repeats as an internal control. So spiking is an external control because this is RNA that you add into the samples. Allele repeats are an internal control because you will determine these in the RNA obtained from your original sample. So allele repeats are repetitive sequences in the transcriptome. And you can see these over here. So these are the blue parts that you can see in the figure on the right. And these allele repeats can be used as a measure of the messenger RNA fraction. So you assume that if a certain sample has a higher number of these allele repeats, that also the total mRNA fraction for that sample is larger as compared to another sample. So here you can see this explained in an example. So for 3 genes, for gene A, you can see that the number of allele repeats is the same in both sample 1 and sample 2. So both have 2 of these allele repeats. So this means that the ratio between sample 1 and sample 2 is 1/1. However, for gene B, you here see that you have 3 allele repeats for sample 1, but only 1 in sample 2. So here the ratio is 3:1. For gene C, the situation is the opposite. However, the total number of repeats between sample 1 and sample 2 is the same. So both in this simplified example have 6 repeats. So overall, this means that differential expression of a small number of genes will not influence the overall repeat abundance in the transcriptome. So this means that even though some genes are more highly expressed in one sample compared to the other, there is probably other genes that show the opposite pattern. And this will level each other out. So what this means is that if the number of these repeats strongly differs between 2 samples, this most likely means that the total mRNA fraction is also different between these samples, and you would then normalize against this. However, this method is difficult to use for unknown genomes. Because in that case, maybe not much is known about the presence of these allele repeats. And also, this does not take into account the purity and integrity of the obtained RNA. So also these methods, both using an external and this allele repeat internal control, is not ideal. Now, what would then be a good normalization method? Well, correct normalization would take into account different things. First of all, sample size, also concentration, quality of the RNA. So this means both the RNA purity and the RNA integrity. It also takes into account reverse transcription efficiency, so how efficient is the cDNA synthesis, and it will also take into account the presence of PCR inhibitors in the samples. So the best normalization method that you can use is normalizing against the expression of internal reference genes. So this means that in your cDNA samples, you will not only measure the expression of your so-called genes of interest, but you will also measure the expression of certain reference genes. And what are now reference genes? Well, good reference genes are genes that show a stable expression across different tissues, across different growth stages, across samples exposed to different stress conditions. So basically, their expression should be relatively stable, irrespective of all these things, such as tissue, growth stage, stress condition, and so on. So let's say you perform an experiment with plants which are exposed to a certain stress condition. This could, for example, be cadmium stress. Then good reference genes would be genes whose expression is not affected by cadmium exposure. And then if you would still see a difference in expression of these reference genes between different samples, this would most likely indicate that there are some technical differences between those samples. So for example, differences in the amount of PCR inhibitors, differences in RNA concentration, and so on. Now, which genes would be good reference genes? One type of genes that are often used as reference genes are so-called housekeeping genes. And housekeeping genes are genes that encode proteins that are involved in basal cell activities or structural cell components. And these are assumed to have the same expression level as we already mentioned, in different tissues, at different developmental stages, under different growth conditions, under different stress conditions, and so on. So these could, for example, be genes that are involved in energy production, and defense against cell death, and the biosynthesis of macromolecules such as DNA or proteins, and disposal of waste. So these are all very important, crucial functions for cells. And as a consequence, we expect those genes to be constantly expressed at the same level, even when an organism or a cell is exposed to certain stress conditions. And at the bottom here, you can see some examples. So, for example, the 18S ribosomal RNA, GAPDH, elongation factor 1 alpha, and when you look in the literature, you will find many more. But the question now is, are these housekeeping genes always good reference genes for normalizing qPCR data? So many researchers standardly use such housekeeping genes to normalize their qPCR data. However, what they sometimes forget is that the assumption that housekeeping genes are good reference genes, so that they have a very stable expression, that this is based on northern blots. And as you might remember from the first lecture in this series, northern blots generally have a much lower resolution than qPCR, which means that if there would be small differences in expression, for example, after exposure to stress conditions or in cells or tissues of different developmental ages, then these differences will most likely not be detected using a northern blot. However, housekeeping genes still might be good reference genes, but in that case, it's very important to verify that their expression is indeed stable under new experimental conditions. But this is something that researchers often do not do. So they will just assume that a certain gene, a certain housekeeping gene is a good reference gene, and they will just use it for data normalization without checking whether it actually is a good reference gene. So it's very important to test your reference genes under your experimental conditions, so in the experimental setup that you are using. So housekeeping genes can be good reference genes, but this is not necessarily always the case. So here, last sentence is very important. A reference gene is an endogenous control gene that is evaluated in every experimental condition. So if you are, for example, always performing experiments in plants exposed to cadmium stress, you might know from experience that a certain gene is always a good reference gene. However, if in the next experiment you will start investigating responses of plants to heat stress, then you would have to check whether this specific gene is also a good reference gene under heat stress conditions. So that's always very important to keep in mind. Now, how can you then select good reference genes for real-time PCR? Well, the golden standard is to always use multiple reference genes. So you would always use a combination of 3 or more reference genes. So never use only 1 or 2, always use 3 or more. And you should always validate the fact that they show only very minimal variation in expression levels between your experimental conditions. To select these reference genes, we typically use this flow chart here. So first of all, you will need to select genes that you want to validate. So you will need to look for genes which you think could be good reference genes under your experimental conditions. How could you find these? Well, you could, for example, look into literature. If someone else performed a qPCR experiment in similar experimental conditions, then you could have a look at which reference genes this person used. Of course, if you're performing similar experiments frequently in your research, then you could also have a look at your own previous experiments and see which reference genes were suitable in those experiments, because of course there is a high likelihood that they will also be good reference genes in your new experiment. And of course, housekeeping genes can also always be considered because, as I mentioned, they're not always good reference genes, but they often are. After then selecting potential reference genes, you then have to validate them. And validating means that you will actually measure their expression in your set of samples. And the golden standard is to measure the expression of at least 10 candidate reference genes. So you should measure 10 or more. And of course, you need to do this in the same cDNA samples that you will use to measure the expression of your genes of interest. And then you can use an evaluation algorithm to tell you which of these 10 or more reference genes are the most suitable to use to normalize the data from this specific qPCR experiment. And there are different algorithms available. For example, there is GNorm, which is very frequently used. Norm Finder is another one, and GrayNorm is a relatively new evaluation algorithm, which has been developed in our research group at Hasselt University. And I will also explain you a bit more about the principle behind this algorithm later on in the lecture. And then the last thing that you need to do is also you need to revalidate the chosen reference genes in related or repeated experiments. So if you have performed one experiment and found a set of 3 or 4 reference genes to be suited for normalizing that specific qPCR experiment, when you repeat this experiment, you will need to again validate that, again, these genes show a stable expression across their experimental conditions. Often this will be the case, but it's very important to always validate this again. And here you can also see this flowchart and visual scheme. So again, with these 3 steps. So first of all, you will select potential reference genes. And here you can see different possible sources. So different possibilities of which genes you could consider as reference genes. Then you will validate these by measuring them in your cDNA samples. And then for new experiments, you always need to revalidate them. So then in the next set of slides, we will go through the principle behind the GrayNorm algorithm that I just talked to you about. So the goal of this algorithm is to determine which reference genes are best suited to normalize the data obtained from a particular qPCR experiment. So here you see the title of this publication describing the GrayNorm algorithm. So the title is Reliable Gene Expression Analysis by Reverse Transcription Quantitative PCR, Reporting and Minimizing the Uncertainty in Data Accuracy. So this publication will also be made available to you. But then the first question you could have, of course, is we mentioned here minimizing uncertainty in data accuracy. But the question is, where does this uncertainty then come from? And I will try to illustrate this using the example here. So in this graph, you can see the expression levels of the RBOHF gene. RBOHF is a pro-oxidative gene. And this expression was measured in Arabidopsis thaliana plants that were exposed to different concentrations in the micromolar range of zinc. And the data that are shown here are normalized data. So after data normalization using certain reference genes, but no further information is given. So here on the x-axis, you can see the different concentrations of zinc that the plants were exposed to. And on the y-axis, you can see the relative expression of this gene. So the expression in the control samples that were not exposed to zinc is 0. And then the expression levels in the plants exposed to 100, 250, and 500 micromolars of zinc are shown here, and they're all clearly higher than the expression in the control plants. But these are only the normalized data, so the non-normalized data are not shown here. So then you can wonder, are these data now accurate? And we will discuss this now in more detail in the next slides. And then hopefully with these slides, I can convince you that it's actually uncertain that these data are indeed accurate. So in this case, and I will always work with a certain example. So we have two samples. Sample 1 is a cDNA sample obtained from controls. So in this case, we could say control Arabidopsis plants. And sample 2 is a cDNA sample derived from plants treated with a high zinc concentration. Now, as we already mentioned several times, there will be technical variation. This technical variation could be, for example, differences in the RNA concentrations of the RNA samples that the cDNA was generated from. There could also be differences in RNA purity or integrity, and there could be differences in the presence of PCR inhibitors. And this is, of course, sample specific. It's possible that, for example, in sample 1, there are more PCR inhibitors compared to sample 2. So each sample has its own technical variation, which we here name T1 for sample 1, and T2 for sample 2. Then what we will do is we will measure the expression of our gene of interest. And this expression, we show it as a so-called RQ value, and RQ stands for relative quantities. And in the demo on data analysis, it will become more clear how this is specifically calculated. But the calculation is based on the CQ values that you obtained during the qPCR reaction. So here you can see the RQ value for the gene of interest in sample 1 and the RQ value for the gene of interest in sample 2. And of course, these are both subjected to their own technical variation. So T1 for sample 1, T2 for sample 2. Now, what you will also do is you will also measure the expression of a reference gene. So also for the reference gene, you will determine the CQ values for each of the samples. But this technical variation, for example, differences in RNA concentration, differences in PCR inhibitors, this will be, of course, the same for the gene of interest and the reference gene, because they're measured in the same cDNA samples. Now, what will happen during the data normalization is that these values will be divided by each other. So as a result, you will obtain a so-called normalized relative quantity, or NRQ value. And this T1 will be divided by T1, and T2 will be divided by T2. So, of course, these level each other out, so that in this case, the data normalization corrected for any technical variation that might be present. And this is, of course, the goal of the normalization. I will make it more clear here with a more specific example. So differences in technical variation could, for example, be related to differences in RNA input. So you isolated RNA from sample 1 and from sample 2. And then from that RNA, you generate cDNA. But maybe due to a technical issue, mistake that you made in the lab, the RNA input for sample 1 was only half that of the RNA input for sample 2. So in sample 2, you used twice the amount of RNA compared to sample 1 in order to generate cDNA. So then the technical variation for sample 1 will be equal to 1. The technical variation for sample 2 will be equal to 2. When you then measure the gene of interest, you obtain so-called RQ values. So here the technical variation is 1, here the technical variation is 2. As I mentioned, the technical variation is of course the same for the gene of interest and the reference gene. So again, number 1 for sample 1, 2 for sample 2. What then happens during normalization is that here 1 is divided by 1 and levels each other out. The same here, 2 divided by 2 also levels each other out. So in both cases, you here get number 1. And this means that the normalization using the reference gene has corrected for this technical variation. In this case, if you would have not performed this normalization, it might have looked as if the expression of the gene of interest was twice as high in sample 2 compared to sample 1. But actually, biologically, this is not the case. The expression of this gene maybe was the same between both samples, but it's just because there was a different RNA input that this made it seem as if the expression of the gene was higher in sample 2 compared to sample 1. But because this technical variation is the same for the reference gene, normalizing against the reference gene will take out this technical variation. So the reference genes correct for sample-specific technical variation. So this is something you can see here again. So technical variation for sample 1 equals 1. Technical variation for sample 2 equals 2. And after normalization, this technical variation will be leveled out. However, the problem with this is that in this case, you assume that reference genes are perfect. So this means that if between different samples, you would obtain different CQ values for the reference gene, that this would only be due to technical variation. So if according to your qPCR results, there is a different expression of the reference gene between different conditions, this would actually be a consequence of technical issues, such as presence of PCR inhibitors, use of a different RNA input, and so on. However, in reality, this is not the case because reference genes are not perfect. Of course, we try to use reference genes that are as close to perfect as possible, but they are never 100% perfect. So this means that in real life, reference genes will also show small differences in expression between different conditions. So in our example, it would be the case that zinc exposure of Arabidopsis plants will also slightly affect the expression of the reference genes. Of course, normally it will not affect the expression of the reference genes as much as that it will affect the expression of the genes of interest, but still there might be a small effect on the expression of the reference genes as well. And this is what we would call biological variation. So biological variation means that inside the plant cells, before you isolated the RNA, there was an actual difference in expression of the reference gene. And the biological variation we denote with a letter B, so B1 for sample 1, B2 for sample 2. You then measure the expression of the gene of interest. You obtain the relative quantities or RQ values for this gene of interest. And you do the same thing for the reference gene. This biological variation, of course, is only linked to the reference gene, because the biological variation, so the actual variation and expression, will not be the same for the reference gene and the gene of interest. Maybe the gene of interest, in our example, this was the pro-oxidative gene RBOHF. Maybe this gene would be strongly increased in expression in response to zinc. But maybe the reference gene has a lower expression after exposure to zinc. Or there could also be an increase in its expression, but a smaller increase compared to the one seen for the gene of interest. What will then happen after normalizing is that you see here that you will divide 1 by biological variation of sample 1, and on the right side you will divide 1 by the biological variation of sample 2. So here an example to make it a bit more concrete. Let's say that in sample 1, the expression of the reference gene is only half as high as in sample 2. So in sample 2, the reference gene is expressed 2 times more than in sample 1. This is, of course, a very extreme example, but we made it in this way to really make the concept clear to you. So here, the biological variation in the reference gene for sample 1 equals 1. For sample 2, it equals 2. You then measure the expression of the gene of interest, determine the relative quantities. You do the same thing for the reference gene. And then when you normalize, what will happen is that to calculate the normalized relative quantities, or NRQ, there won't be any effect of normalizing in sample 1, because here you will just divide by 1, which will of course not have an effect. However, in sample 2, you will divide this RQ value of the gene of interest by 2. So here, normalizing the data will have a very strong impact on the final result, because in this case, there will be a twofold difference between the non-normalized and the normalized data. So what you do in this case is that you inversely impose the reference gene-specific biological variation on the gene of interest. And this is something you do not want to do, because you want the reference genes to correct for technical variation, but you do not want to have any impact of the biological variation in the reference gene. So we now nicely separated them in the previous slides. So we had some slides on technical variation, we had some slides on biological variation, but in real life, of course, there will be both. When you see a difference in CQ value between, for the reference gene between 2 samples, part of this will most likely be due to technical variation, such as differences in RNA input or different amount of PCR inhibitors. But a part of the difference in CQ value will be due to biological variation in the reference gene. So this will be due to actual differences in expression of the reference gene between the 2 samples. So in this case, when we make the calculations, we will correct for the technical variation, because we divide here T1 by T1 and T2 by T2, which means that we will completely take out any technical variation, but we kind of correct too much, because we're also correcting for this biological variation in the reference gene. And this is actually not what we want to do, but we cannot avoid this. So the reference genes correct for technical variation, which is a good thing, but they also impose biological variation on the gene of interest. And this is something you would actually want to avoid. So the problem is that we do not really know which part of the difference in CQ value between 2 samples is due to technical variation and which part is due to biological variation. It might be that it's half-half, so half of the difference is related to technical variation, half is related to biological variation. But of course, the two opposites, let's say, are also possible. One possible assumption is that your reference genes are nearly perfect. So this would mean that biologically, they do not show any difference in expression between different samples. So then all the variation would be technical. So technical is shown in red, biological in yellow. So here you see that almost the entire bar color is red. So when reference genes are almost perfect, differences in CQ value for the reference gene between samples would only be due to technical variation. On the other hand, when you have an experiment of very high technical quality, in that case, all the variation or most of the variation that you would measure in the CQ values of the reference gene would be due to biological variation. And what do I mean with an experiment of high technical quality? Well, this would mean that the RNA concentrations would be extremely similar between the different samples. There would be no PCR inhibitors present. The efficiency of cDNA synthesis is exactly the same between the different samples, and so on. So in this case here, where the reference genes are nearly perfect, that normalization is actually a good thing, because you correct for technical variation, which is ultimately the goal of normalizing the data. On the other hand, when your experiment is of very high technical quality, then normalization is actually not necessary, because then you're only correcting for biological variation in the expression of the reference gene. And this is not the goal of data normalization. So in that case, the non-normalized data would be more correct. But unfortunately, you cannot know. There is no way to find out whether the reference gene differences in CQ for the reference genes between samples would be due to technical or due to biological variation. So here we will have a closer look at these two assumptions. When the reference genes are perfect, there will be no biological variation. So the biological variation in the two samples would be the same. Then here, for the gene of interest, we would of course have the technical variation. For the reference gene, we would have the technical as well as the biological variation. But the technical variation would level each other out, and the biological variation, as we mentioned that there isn't any, we could leave it out of the equation here. And in this case, the normalized data would be the most accurate. On the other hand, when you would have a technically perfect experiment, there would be no technical variation. So we would only have biological variation in the reference gene. So when we then make the calculation, what we see is that the technical variation will level each other out, but this would not even be necessary because, as we mentioned, there is no technical variation. But in this case, we're inversely imposing the variation in expression of the reference gene, so the biological variation, on the gene of interest. And this is not what we want to do. So in this case, actually, the non-normalized data would have been more accurate than the normalized data. So in this case, it would have been better to not normalize the data. But of course, this is something you cannot know. So what the concept behind the gray norm algorithm says is that the truth lies somewhere between the normalized and the non-normalized data. In the normalized data, you corrected for technical variation, which is a good thing. However, you also imposed this biological variation of the reference gene, and that's not what you want to do. So in the non-normalized data, you did not do this. So that's also a good thing, of course. So what we propose is that you perform a so-called gene expression sensitivity test, which means that you consider both the normalized and the non-normalized data, and you perform statistics on both. And in both cases, you should find a significant difference. So If both the normalized and the non-normalized data are significant, only then you can say that you found a significant difference. And what the gray norm algorithm will do is it will select the reference genes for which the ratio of 1 over the normalization factor shows the smallest deviation from 1. And 1 over the normalization factor is the number with which you will multiply the relative quantities of the gene of interest in order to normalize them and obtain the normalized relative quantities. All of this will become more clear during the demo on data analysis. So you can maybe look back at this presentation after also watching the demo, and then this will for sure become more clear. So I will now come back to this slide here with the relative expression of this pro-oxidative gene in Arabidopsis plants exposed to different concentrations of zinc. As I said previously in this graph, you only see the normalized data. And based on these data, you would say that exposure to these higher zinc concentrations increases the expression of this gene. However, when we also show the non-normalized data in black here on the same graph, you would see that there are some small differences between the normalized and the non-normalized data. However, for the plants exposed to 100 and 250 micromolars of zinc, you can still with certainty say that the expression of the gene increases compared to the control. However, for the plants exposed to 500 micromolars of zinc, the normalized data indicate that the expression of RBOHF increases. But according to the non-normalized data, there is no difference in the expression of the gene compared to the control, or even a slight decrease. So in this case, you cannot reliably make a conclusion on or regarding the expression of this gene in the plants exposed to 500 micromolars of zinc. And this difference here between the normalized and the non-normalized data, which is shown in gray here, this is what we call the gray zone. So, for example, in this case, for the 100 micromolars of zinc, the white bar, so the normalized data, would result in a conclusion that the expression of the gene is increased 3 times compared to the control. According to the black bar, for the non-normalized data, you would say that the expression is increased 2 times compared to the control. So you can say with certainty that the expression is increased, but the actual number of the increase could differ. So it's either 3 or 2 or any value in between. So that's sort of our uncertain or gray zone. And because we call this zone the gray zone, the algorithm is called gray norm. So normalization based on this gray zone. So how would you now represent these data in a publication? Well, if you only measured the expression of a limited set of genes, you could show them in a graph, such as a histogram here. Then you would show both the normalized and the non-normalized data, and you would perform the statistics on both, and both should be significant. So in this case, for the 100 and the 250 micromolars of zinc, you can see that both for the normalized and the non-normalized data, the expression is significantly higher than that of the control. So here you could say with certainty that the expression of the gene is increased in response to these zinc concentrations. However, for the 500 micromolars of zinc, the expression is significantly increased according to the normalized data, but there was no significant difference found for the non-normalized data. So in this case, you cannot say with certainty that the expression of the gene is indeed increased by exposure to 500 micromolars of zinc. Of course, the normalized data do suggest that there is an increase, but you should be cautious with interpreting these data and drawing conclusions from them. On the other hand, when you measure the expression of a larger number of genes, what is usually done is to show them in a table. And what you could do in this case is in the supplemental information of a publication, you could provide so-called resolution values. And these resolution values are the 1 over normalization factor ratio, which you would show for each condition. And remember that 1 over normalization factor is the value that you multiply the RQ values for in order to normalize them. So this means, in this example here, you see here a resolution of 1, here a resolution of 0.95, here a resolution of 1.26. So these values do not deviate very much from 1, which means that the normalization will not have a very big impact on the obtained results. However, for this condition here, shown in red, the 1 over normalization factor is 0.46. So this means that on average, the RQ values are multiplied with 0.46 in order to normalize them. But of course, multiplying by a value of approximately 0.5 means that you will divide the obtained value by 2, which is of course a very large effect. And here you would then see in this bottom half of the table the expression of a gene of interest after normalization. Here you see value 1 for the control. In this condition, you see that the value is 0.29 and here it's 0.45. But because here the 1 over normalization factor did not deviate much from value 1, you can say with certainty that indeed in these conditions, the expression of the gene of interest is lower than in the control. However, you could also say that for this condition here, because here the value is 0.34, which is also much lower than the value 1 that you observe in the control. However, in this part of the table showing the resolution, you see that a large part of this effect is actually caused by the normalization. And in that case, you should be cautious with interpreting the data, because this value 0.34 here really mean that the expression of the gene of interest is decreased, or is this just a consequence of the normalization? And can we not be certain about our conclusion regarding the expression of the gene of interest in this condition? So in general, these were the best ways to represent your data. And by showing the data in this way, you are transparent regarding the uncertainty in your data. And then people who read your publication can decide for themselves whether or not they trust the obtained results. And then one last thing that I briefly want to discuss regarding reporting qPCR data are the so-called MIQE guidelines. The MIQE guidelines were first published in 2009 by an international consortium of academic scientists who were strongly involved in the qPCR field. And MIQE stands for Minimum Information for Publication of Quantitative Real-Time PCR Experiments. And these guidelines give a uniform standard for reporting on qPCR experiments. So these guidelines, they provide you with a list of parameters regarding qPCR experiments that you should report on in publications showing qPCR data. And here you can see an example of this checklist. So I will not completely go through it, so I will not discuss each of these parameters, but things that should, for example, be specified in a publication on qPCR data are, for example, what is the source of the sample that you obtained? How did you preserve the samples prior to isolating RNA from them? How long were the samples stored? Which method did you use to extract the RNA? How did you determine the RNA concentration and the integrity? Then regarding the qPCR assay itself, which primers did you use? How did you test the PCR efficiency? Which protocol did you use to generate the cDNA? Which software did you use for the data analysis, and so on? So this is an entire checklist that you should fill in, and then you can add it as supplemental information to publication in order to be transparent on the way in which you conducted a qPCR experiment. But a publication in Nature Methods in 2013 showed that unfortunately many publications that report qPCR data still do not adhere to these MIQE guidelines, which is of course a very unfortunate thing, because the reason of these MIQE guidelines is for people to be more transparent on the experimental procedures that they used. And this is something that we can only encourage. So if in the future you ever publish qPCR data, please keep this in mind and please do adhere to these MIQE guidelines. So to conclude this lecture, I want to give you a few take-home messages. So as we saw today, qPCR is a very sensitive method, which is of course a nice thing because we can detect small differences in expression between samples, but this also means that it's also highly sensitive to technical variation. So throughout the qPCR workflow, technical variation should be minimized. And it's essential to normalize qPCR data with reference genes that you validate for each experiment. And it's also very important to report uncertainty in qPCR data accuracy, for example, by showing both normalized and non-normalized data. And when publishing qPCR data, you should always adhere to these MIQE guidelines and fill in the MIQE guidelines checklist on all the parameters or all the experimental details regarding the qPCR experiment that you performed.